Dear colleagues, my name is Schiesari. I am a professor of environmental management at the University of São Paulo, and today my presentation will be about the ecology of sugarcane fields. The debate regarding the environmental benefits of a large-scale adoption of biofuels has heavily emphasized reductions in greenhouse gases, neglecting other highly relevant forms of environmental change, such as biodiversity loss. However, we know that biodiversity is sensitive to a variety of physical disturbances, both in magnitude and frequency, and to a variety of chemical stressors that characterized anthropic land uses. So it is very important that we understand what is the biodiversity value of sugarcane and how to improve it faced to other forms of land use. We proposed to investigate the environmental drivers and the demographic mechanisms by which species are added, replaced, or deleted across gradients in land use intensification. We have emphasized freshwater ecosystems, in particular small freshwater ecosystems, both because they allow sufficient replication for statistical power and because they are inhabited by species that are amenable to experimentation in lab, mesocosms, and field, so that we can experimentally try to tease apart the factors that are driving changes in community structure across this gradient. Our model freshwater communities are dominated by algae and macrophytes as primary producers, as tadpoles as amphibian larvae as detritivores and herbivores, and a variety of predators, which include insects such as dragonfly larvae, beetle larvae, aquatic bugs, and fish. We have conducted a comparative sampling surveys across gradients in land use intensification and found that sugarcane fields are impoverished in biodiversity, both of vertebrates and invertebrates, when compared to cerrados and sometimes also pastures. However, what we also found is that there are species that are found throughout the gradient and therefore are capable of colonizing and inhabit and are exerting their function in sugarcane fields. Focusing on amphibian larvae, what we have found is that Dendropsophos minutus, Dendropsophos nanus, Sinax, Physalemus natereri, Physalemus cuvieri, and Leptodactylus fuscus are found throughout the gradient. Actually, if we plot the frequency of occurrence, that is the percentage of water bodies in which they occurred, and their per unit area biomass, we found that these species are not being significantly affected by land use, at least by means of this criteria. So Nayana Cândido went over and measured and weighed thousands of these tadpoles. And with this, we have calculated the body condition index, which is the residues of the biomass as a function of developmental stage. And what we have found is that tadpoles in sugarcane fields are actually as heavy as those found in pastures and native habitats relative to their developmental stage, which suggests that they might be doing well. Furthermore, some individuals found in sugarcane fields are very large relative to their stages, which prompted us to understand what were they eating and what was their relative nutritional value. So João Barcelos Leite, my student, selected 3 of these species and took them to the laboratory, where he fed them the decomposing leaf litter of cerrados, pastures, and sugarcane fields. And what he found is that the decomposing leaf litter of sugarcane is of sufficient nutritional quality for permitting tadpole growth, and this happens because sugarcane leaves are comparatively tender and therefore decompose fast, yielding high-quality detritus. However, what we also found is that there are occasional faunal die-offs in ponds adjacent to sugarcane field, as witnessed in this particular example. Therefore, there must be episodes in which populations could be entirely wiped out from sugarcane fields. So we hypothesized that this could be due for the employment of agrochemicals. And we manipulated pesticides and fertilizers such as ammonia and vinasse, exposing a variety of freshwater organisms to these compounds at realistic dosages. And what we found is that both pesticides and fertilizers can cause significant mortality and sublethal effects, including growth, development, and reproduction impairment, in organisms as diverse as amphibian larvae, dragonflies, midges, crustaceans, and fish. To better understand the ecological, ecotoxicological, and biogeochemical implications of land management for bioethanol production, we moved to large-scale experimentation and actually converted extensive pastures into intensive pastures and sugarcane fields. This is one of our experimental sites in Brotas, showing 15 0.25 hectare plots. The 5 plots in the front are extensive pasture, the 5 plots in the middle are intensive pastures, and the 5 plots in the back are sugarcane fields. Now this set of experimental plots received 15 9,000 liters and 18 3,500 liters mesocosms, which are constructed freshwater ecosystems. The realism of these constructed freshwater ecosystems can be verified by comparison with a natural terracing pond found inside of sugarcane fields and that contain a variety of insects and amphibian larvae. So these experimental plots were subject to conventional land management, including harvesting, fertilization with vinasse, and application of the insecticide fipronil and the herbicide 2,4-D. As one might expect, the physical-chemical changes in these mesocosms were steep, in particular after the application of vinasse, which is represented by the red dotted line. So dissolved oxygens dropped to 0 in sugarcane mesocosms. In turn, conductivity levels increased fourfold, to a large extent by the increase in carbon, nitrogen, phosphorus, and potash, which, after a few weeks, promoted a bloom in phytoplanktonic algae. Biodiversity responses were varied. At least in terms of abundances, colonizing amphibian larvae seemed to be irresponsive to land management. By contrast, insects, and in particularly sediment-dwelling and benthic insects, suffered strong consequences of land management, in particularly vinasse and secondarily fipronil. We can analyze the trajectories of the communities of these 3 different types of land management by means of an ordination technique. What we see here is that vinasse and fipronil promote strong divergence in sugarcane communities. However, over time, sugarcane communities converge to those of pastures via the arrival of new migrants. What we found then is that the biodiversity of freshwater ecosystems depends on the management of the surrounding agricultural matrix. We infer then is that improvement of land management practices can promote an increase in the biodiversity of sugarcane fields. However, we know that even freshwater ecosystems are not entirely isolated in space, but are connected to other freshwater ecosystems by the dispersal of organisms over the terrestrial landscape. So these networks of freshwater ecosystems are called meta-ecosystems, and they harbor meta-communities of aquatic and semi-aquatic organisms. The consideration of meta-ecosystem dynamics tells us that, actually, spatial context can be important in influencing, in modulating the response of biodiversity to land management. To better understand these problems, my student Rodolfo Pelinson led a field experiment in which artificial ponds were created at 30, 120, and 480 meters from a stream and its floodplain, which function as a source of colonists dispersing over the terrestrial landscape. So a view of these rows of artificial ponds or mesocosms can be seen here. So these ponds were colonized by our model freshwater communities. But not only spatial isolation was manipulated, but also it was fully crossed, with a simulation in scheduling dosage of agrochemical intensification that follows the conversion of cerrado into pastures and pastures into sugarcane fields. 1/3 of the ponds receive no agrochemicals and therefore represent, quote unquote, our cerrado ponds and communities. 1/3 of the ponds received weekly additions of nitrogen, phosphorus, and potash, simulating the gradual dissolution of granulated fertilizers and therefore represent, quote unquote, pasture ponds. Finally, 1/3 of the ponds receive not only the weekly additions of NPK, but also a single pulse of fipronil and 2,4-D, and therefore represent our quote unquote sugarcane ponds. We then follow community composition and structure and ecosystem function over time. So the results will be shown by means of ordination plots. So in these ordination plots, each symbol represents one community. There are 45 communities because there are 45 ponds. And these different symbols are joined in ellipses that represent each of the isolation treatments. So what we see right away is that spatial isolation strongly influences community structure, and that distances as close as 120 meters are sufficient to cause strong effects on community structure. And this happens because of dispersal limitation. Not all organisms from the source wetland are capable of reaching these ponds that are 120 meters. Once agrochemical application starts, then fipronil is found to promote a major change in community structure, with communities of sugarcane pods, quote unquote, in the lower left, strongly diverging from the other treatments because of the deletion of insect populations and dominance by amphibian larvae. The subsequent manipulation of 2,4-D had no further consequence to community structure, and after 120 days, spatial isolation and agrochemical still were important and still influenced community structure, even though the community differences become more and more blurred over time. In conclusion, as other agricultural crops, sugarcane fields are impoverished in biodiversity relative to forests and cerrados. Yet, sugarcane fields harbor a subset of tolerant generalistic species. These species are either highly mobile species that visit or traverse sugarcane fields, or fast-developing species that colonize sugarcane fields until physical disturbances and chemical stressors remove their populations. Now, recolonization of sugarcane fields depends on organismal dispersal, which is inversely related to spatial isolation. In fact, distances as small as 120 meters into sugarcane fields are sufficient to limit species colonization. Now, despite significant recent advances, if sugarcane is to fully satisfy its claim as the leading feedstock for the environmental sustainability of fuels for transportation, biodiversity must play a key role in the agenda of the sector. That is, the biodiversity value of sugarcane fields can and should be significantly increased, and there are different ways by which this can be achieved. First, it could be improved by improved land management practices. And second, it could be improved by the protection, restoration, and conservation of both in-field and out-of-field habitats in landscape management. Thank you very much.